Hey everyone, so this is a screencast review for meiosis. Before I get into meiosis, actually, I wanted to ask you a different question. And that question is, have you heard about that hot Swickley baseball team and how well they're doing? If you haven't, I suggest you go out and check out some of their games because they're doing really well. And you're going to want to say that you've been there, been to some of those games. They're doing really well. For the baseball players amongst you that are hopefully listening to this, I wouldn't, rest just on that. There's a lot of games to be played, right? So uh, keep at it. But so far so good. So nice job. This screencast is dedicated to them. Okay, so this meiosis screencast goes out to them. All right, first thing you have to do though when considering meiosis, there's a lot of vocabulary that you have to consider with meiosis, right? The first thing I want you to understand is this idea of what a karyotype is. So a karyotype is a complete accounting of all of the chromosomes that you would find in a cell. Okay, so all of them. Now, human beings have a total of 46 chromosomes in their body cells. So that's like not their sex cells, it's their body cells. So this karyotype right here contains 23 pairs. So you got 22. This X and Y stand for the 23rd pair. So 23 pairs. And that equals 46 total. Now because you have 23 pairs, one pair comes from dad and one pair comes from mom. Okay, so you get two pairs, 23 chromosomes in a pair, 46 total. And the karyotype basically shows you all of those chromosomes. So these are artificially colored to show the differences between each chromosome, right? So this big yellow one is pair number 1. This big orange one is pair number 2. And you could literally go through and label, okay, so let's say that the ones on the left are from dad. This is from mom, dad, mom, etc. Okay, so if we take these two, this pair, each of these individual chromosomes, so this one and this one, have a special name, so that's homologous pair. So this is homologous pair number 5, okay? Again, one from dad and one from mom. So stick with me at the vocab. Homologous pair, karyotype, uh, and the number of pairs of chromosomes. So two pairs, 23 pairs, excuse me, two sets. So this should say two sets. Two sets of chromosomes, 23 chromosomes in a set, therefore 23 pairs for 46 total. This is important when considering the human life cycle. At different stages of the human life cycle, various different cells are being made. So there are two types of cells. There are haploid cells and there are diploid cells. Haploid cells have one set chromosomes. Diploid cells have two sets. Okay, so in the case of humans, diploid cells have 46 total, and haploid cells have 23 total. Okay, these different types of cells are made through different processes. So generally for diploid cells, these cells are created, they're produced by a process called mitosis. You already know about mitosis and how a cell can duplicate its DNA and then divide it to end up with matching diploid cells. The process of meiosis creates haploid cells that have one set of chromosomes, so 23. Here's the thing with that. So you need to start life with haploid cells, okay? So here in this life cycle, we have diploid adults, okay? All their body cells, so cells in their arm, brain cells, liver cells, all their cells are diploid for the most part, with the exception of cells in their body that create gametes, right? So for each sex, for the females in the ovaries, for the male intestines, there are going to be cells in that region that produce haploid cells. Okay, so we're pointing here to a haploid cell in the egg, here a haploid cell in the sperm. Note that there are 23 chromosomes in these. It's important to use meiosis to make these haploid cells because if you recall, humans as an organism are for the most part diploid. All of our tissue for the most part is diploid. That diploid state is restored during fertilization. So when that sperm enters the egg and fertilizes it, you restore diploidy. Okay, so you're once again a diploid. 23 from mom plus 23 from dad equals 46. So you're restoring that diploid state. Okay, so now you're looking at a diploid zygote right here. It's 46 chromosomes. And that one single diploid cell is going to go on to divide and create this amazingly complex multicellular organism, trillions of cells, known as a human being, right? The process by which these diploid cells multiply, though, is different. If you remember over here, we're creating haploid cells through meiosis, but to double our diploid cells, we're going to go back to mitosis, which you've already, the process you're already familiar with, right? So this one cell becomes 2 through meiosis, the 2 cells become 4 through another round, the 4 cells become 8 through another round, until you're this multi-cellular complex organism known as a human. Here's the thing, there's some other vocabulary that you can use to denote what is diploid and haploid. So I will on the test ask you what an organism's haploid number is, and I will denote it by N. Okay, so I'll ask you what's the N for a certain organism. Okay, I will also ask you what the diploid number is, 2N. That 2 is there to convey to you that there's 2 sets, right? So it's twice as much chromosomes. So for example, again, in a human being, the N, the haploid number, is 23 because there's 23 chromosomes in that one set. Same thing for sperm, they're both haploid. And again down here you'll see the 2N for humans is 46. That's those 2 sets coming together, so 46 total chromosomes. Okay, moving on. It's important to understand a pretty significant distinction between mitosis and meiosis. In mitosis, you're basically taking a cell that has 46 chromosomes. You are then going to double it. So you're going to make 92 sister chromatids. Okay, and then you divide the process of mitosis to get 46 again. But you have 2 cells now, so you get 2 at 46, whereas you started with 1, now you get 2. That's mitosis. You're basically keeping the diploid state intact. It's a little different for meiosis. Meiosis, if you did the same thing, 46 to 92 down to 46, we haven't created any haploid cells. Remember, 46 for a human is still diploid. It's still 2N. And that's what this figure is showing here. If you start with a diploid cell and you duplicate DNA and you divide once, you're not getting technically down to that haploid state. I know that this says N right here and right here, but it's not technically N because you're still going to have sister chromatids attached. And we'll get to that a little bit later. But suffice it to say that although you've divided once, you're still at a state where you're not down to a single chromosome. You would need to divide one more time to get it down to a state like this. Okay, so my whole point is to get to haploid, you need an extra round of division. You can't stop at 46 here. You have to once again divide, and what you'll get is 4 cells, each with 23, which is one set of chromosomes, so 23 total. Okay, that's why you have in meiosis not just 1 round of division, but 2 rounds of division. Okay, whereas in mitosis, just this 1 round is fine. Okay, so meiosis, 2 rounds of division to get to that haploid state. Yet another distinction between mitosis and meiosis. A very important one. You need to know a little bit of additional vocabulary. Actually, you don't. I've already introduced you to sister chromatids in mitosis, and I've already introduced you to homologous chromosomes in the beginning of this. So the first round of meiosis is going to separate these homologous chromosomes. Okay, so that first round separates those two. Now, the second round is going to separate this, and it's going to separate this. So this goes this way, this goes this way, this goes this way, this goes this way. For, once again, 1, 2, 3, 4 chromosomes. So understand, homologous chromosomes separate in the first round of meiosis, and then these sister chromatids separate in the second round. That's a big difference between mitosis and meiosis, because in mitosis, homologous chromosomes do not pair up like this. They only stay together through a process of crossing over, what we'll talk about in a little bit later, in meiosis. That doesn't happen in mitosis. So just keep that vocabulary in mind as well. Let me walk you through this process really quickly. Just like in mitosis, meiosis is preceded by a round of interphase. So this is where your DNA is duplicated. No big deal. We already know that. The only difference, well, not the only difference, but the big difference occurs in prophase 1, okay, where you form a tetrad. A tetrad is basically 2 homologous chromosomes, so in blue here from dad and red here from mom, homologous chromosomes that are duplicated, so they have 2 sister chromatids. They hang out and stick together, forming a tetrad. So if I blow this picture up a little bit, it's going to look something like this. Okay, this homologous chromosome from dad, this one from mom. It's forming a tetrad. 4. 1, 2, 3, 4 sister chromatids join together to form a tetrad. That's a big difference. They don't do that in mitosis. This only happens in meiosis. Okay, so they come together. Everything else in prophase is the same. I'm not going to spend the time to go over that since you already know it. Metaphase is essentially the same. They line up along this metaphase plate right here. Anaphase 1 is a little different because instead of separating sister chromatids at this phase of division like you do in mitosis, instead you're separating these homologous chromosomes. Remember, this pair right here is now being separated. Okay, so now you have 2 sisters going this way and 2 sisters going this way. The other thing, as I mentioned before, with this tetrad over here, you have a spot of overlap right here. Okay, so this spot of overlap is where crossing over occurs. So literally you'll have some DNA from this sister chromatid cross over to this one, and vice versa. There'll be DNA exchange from here to here. Okay, so that's going to create what's called genetic recombination, which you can also see in this picture here. This blue chromatid here now has a piece of red. This red chromatid here now has a piece of blue. That's been exchanged because in the tetrad, there's crossing over. There's literally touching of the DNA and exchanging of that genetic material. Likewise, over here, some of these red chromatids have obtained some blue. Because in the tetrad, the DNA crosses over, getting you, creating new genetic variations. Now I have to go really fast because I only have a minute, but suffice it to say that everything else from here on out you know. In telophase, the cell goes back to normal. Cytokinesis, the cell splits, but we're not done just yet. In this next slide, which is called Meiosis II, essentially everything that you know about Meiosis I is going to occur. Now, we are going to separate sister chromatids in anaphase. Okay, that's the only difference. Everything else precedes, just as in mitosis. Now I'm out of time for this screencast, but we'll go over that again, okay? So I hope that helps.